Hello students. In this episode, let's brief a little on Mendelism. The greatest contribution to the field of genetics is from Gregor Johann Mendel. He worked exclusively on pea plants, the Pisum sativum. Mendel, basically a mathematician from Austria, got interested in biological sciences. At University of Vienna, he worked on Pisum sativum, that is hybridization experiments on the garden pea plant. And during this venture, he presented the paper, The Experiments in Plant Hybridization, in the journal Annual Proceedings of Natural History Society. Students, in spite of his exorbitant work in genetics, lest did the genetic world accept his principles. However, in the year 1900, Mendel's postulates were rediscovered by three scientists in different corners of the world. To begin with, Hugo de Vries from Holland, who worked on evening primrose. Karl Correns from Germany, who worked on maize plants. And Erich von Teschemark from Austria, who worked on other flowering plants. So these three scientists together are responsible for the rediscovery of Mendelism. With that, let's move on to the material that Mendel selected for his studies on genetics. He worked on the Pisum sativum plant. The reason he did was that it was an annual plant which completed its life cycle in just 3 to 4 months. It was a Pisum plant which had many contrasting traits, and it was naturally self-pollinating. These were the three important reasons why he selected Pisum sativum. Mendel thus quantitatively analyzed the inheritance of qualitative characters. And very important to note here was hybrid cross. Monohybrid cross is a cross that traces the inheritance of single character. Here we consider a pure tall plant. Observe that pure tall plant is got both alleles dominant T, is crossed with a dwarf plant which is a recessive with two recessive alleles t and t. These produce their gametes. The tall plant produces gametes, all of them containing the allele T, and the dwarf plant produces the gametes, all of them containing t. And when they fuse to produce a F1 generation, which is a tall plant, the F1 individuals have two alleles T and t. The F1 individuals further are capable thus producing two types of gametes, T, 50% of them containing T and other 50% containing t. Now, we consider one of the F1 individual as female and the other as male, and selfing is performed. The selfing is put on a Punnett square first given by Reginald C. Punnett, where we take the male gametes along the x-axis, that is horizontally, and the female gametes along the y-axis, that is vertically. And this is the combination we get. We get T, we get T, small t, as well as small t and small t. So tall to the ratio of dwarf was 3. That is, capital T is tall, as well as T, small t is tall. Whereas the dwarf is only 1, that is small t and small t. Thus, in monohybrid cross, Mendel proved the law of segregation. That is, in a hybrid, the factors for a character segregate during gamete formation, where F1 generation, which contained T and small t, produced two types of gametes, T as well as small t. We'll get back to you with dihybrid cross in our next episode.